There. Is it working? Hopefully it's working. Welcome everyone back. And I'm very happy to have the seminar back and we can start off the semester. And we're going to be starting off the semester with a repeat offender. Not the first and hopefully not the last one. Chris, who's already given a talk here a little while ago, and we will be very interested to hear what he has to say today about microbial cross-feeding and coexistence. So I actually won't take up any more of your time. Chris, please go ahead. Take it away.
All right. Thanks, Yuri and the organizers for the repeat invite here. So this story goes back 10 years to a time, well, a similar mood to today where we were desperately looking for funding for the lab. And we started to look in some unconventional locations, one of which was DARPA, which is the research agency of the defense in the US. And normally not where you think you're going to find funding for theoretical ecology, but they actually have a program in biological sciences. And in particular, there was a program that came across our radar called BRICS, Biological Robustness in Complex Settings. And the idea here is that we want to be able to engineer microbial communities to go out and do things for us in the real world. But to do that, they need to be robust, they need to be safe. So that was the program. Now, most of the call for program for projects here, you actually had to do something. You actually had to synthesize some microbes, which I'm not capable of doing. But there was buried in the RFP, there was one little bit, Advanced Studies 5, which is for a smaller chunk of money, but they needed better theory for microbial communities. And I thought, okay, we can do that. So we put in a proposal. We weren't sure exactly what they were looking for, so we threw in a bunch of things. But one thing that seemed relevant to our interests was this idea of, basically we proposed that traditional ecological theory that's been developed for macroscopic organisms is focused on competition, predator-prey interactions, but little theory has been developed for this type of syntrophic interactions that are important for a lot of microbes. And they bet. And so what kind of syntrophic networks am I talking about? So we considered two in our project. One was what we call processing chains, where there's a series of consumers that each basically take a bite off a resource and then excrete some leftover, which are used by the next organism. That's what we call a processing chain. But we really focus mostly on more of a symmetric interaction known as cross-feeding. So this is a resource explicit type of mutualism between species. And so today I'll talk about three papers that we have on this cross-feeding interaction. The first two came from our DARPA project, and the third is a recent collaboration that just came out. And this picture is a little bit misleading because the two with actual organisms are pure theory, and the one with the little cartoon actually has an experiment. So anyhow, so getting into it, what we consider is this type of food web module where we have two species of microbes and they're interacting through two essential resources, R1 and R2. And what you can see here is that species one needs resource two to grow, but it can make its own resource one. Okay, so it can synthesize resource one for itself. So we can think of these resources here as something like amino acids and synthesize enough for itself, but also excrete some in an environment. So species two, who requires resource one but can't synthesize it itself, can pick it up, and then species two excretes resource two. So this is the type of cross-feeding interaction. This is the motif that's going to be central to this talk. And so if we put this in model form, we get a kind of resource consumer model, not too dissimilar from, standard resource competition theory. But now we have this extra source term. So not only do consumers deplete resources, they also synthesize them. So this S is the rate that a species one synthesizes resource one, and similarly for resource two. And so we want to analyze this model. And because, we're trained a little bit in the resource competition theory, we want to, try and put in the graphical approach popularized by Dave Tilman, John Chase, Matthew Leibold. So here's a case where we have our two consumers, and they don't actually produce for each other, they just produce for themselves. So their zero net growth isoclines, or zingies, are this horizontal and this vertical line. So species two requires resource one, so it's this vertical zero net growth isocline, whereas species one requires resource two, so it's this horizontal zero net growth isocline. And their consumption or impact vectors are just at these right angles. So going back to Tilman and Chase and Leibold, we can map out the outcome of interaction between these species, and they don't really even interact, right? So as long as each species has its minimum resource requirement met, the two species can coexist. Okay. So now we want to introduce the cross-feeding, and we'll do that by turning on the synthesis and excretion rates. So set S greater than 0. So that doesn't do anything to the zero net growth isoclines, but what it does is it rotates these impact vectors outwards. So now you see that species one is consuming resource two, but it's actually producing resource one. So that's why this points down to the lower right, and vice versa out here for the other species. And so the impact of a little bit of cross-feeding is it expands the region of coexistence. So for a supply point that's in this little wedge here, species one couldn't last on its own, but in the presence of species two, thanks to cross-feeding, its realized niche is expanded through this mutualism. All right, so now let's just crank it up just a wee bit further and have more cross-feeding going on. So what this does is that it further rotates these impact vectors. And what's different about this case, it seems mostly the same, except for this little region down here. So where there's low external supply or even no external supply of these cross-fed amino acids, then we have now two possible outcomes. So either both species go extinct, or as we'll see in a second, they blow up to infinity. So it's, kind of our stereotypical picture of obligate mutualism down here in the corner. This is showing the time dynamics of our two species, and symmetric, so you don't see the other guy. He's hiding right behind. If we start them with low initial abundances, indeed, they go extinct. But if we start them above some threshold, then they're blowing up to infinity, in a Bob May sort of way. So to summarize, to make this easier to see, we projected this onto the N1, N2 subspace, that is the abundance of our two species, using the quasi-steady state approximation that MacArthur popularized. And what we see are isoclines that are pretty much identical to the Lotka-Volterra model of mutualism, where we have a stable equilibrium at 0, 0, an unstable equilibrium, and then this funnel off to infinity. So even in the absence of supply of these amino acids, we can get the system to persist or blow up through this bistability. Now, we don't like populations blowing up to infinity, actually. And so we know a few different ways to fix it. In our cross-feeding situation, we considered maybe limitation by precursors to those amino acids. But in the end, we settled on limitation by another resource. In this case, this would be something like a macronutrient like glucose. And so this ultimate limitation by energy or glucose is enough to prevent the system from blowing up to infinity. Here's our model when we add this third resource. So R3 throughout the talk is going to be the the glucose, when you think of as what ultimately limits the whole system. And it's consumed like normal resources, whereas the amino acids are excreted. And to determine the growth rate of our two species, we treat them as purely essential resources, so we use Liebig's law of the minimum. But for species one, it's the minimum of amino acid R2 and glucose, whereas for species two, it's amino acid R1 and glucose. We wanted to map these into zero net growth isocyanates, but now it's in this three-dimensional space. So the vertical axis on these plots is the glucose. And so what you see is the zero net growth isocyanates are these L-shaped planes of typical for essential resources, but they're turned in different directions depending on which amino acid you need. Okay. And we consider a couple cases. This is a little bit degenerate, but if the two consumers are equal competitors for glucose, then their isoclines intersect in a plane down here, and equilibrium, they're both limited by glucose. Whereas if we were to make one of the competitors just a little bit better glucose competitor, then this, it breaks that degeneracy. The system goes to equilibrium down here, where the better glucose competitor is limited by the amino acid that's partner provides, whereas its partner is limited by glucose. Okay. This is the same thing, but now just project into the N1, N2 space using the quasi-steady state setup. And again, these two scenarios. So here we have this kind of slightly weird, degenerate, neutral set of equilibria. But if one species is a better glucose competitor, then that breaks that, and we get this, unique equilibrium, although still with the bistability. Okay. So this is great, but why create, why make amino acids for your partner? It's kind of a waste. What if a cheater came along who produced neither? So that's what we wanted to investigate. So because we want to add a third species to this, it's going to get complicated. So we basically took a slice through this left-hand case of what we call symmetric mutualists. So if we assume the two mutualists are perfectly symmetric and the environment's perfectly symmetric, then all the dynamics happens along this red line. And we can reduce this to a one-dimensional system where on the x-axis we've got the abundance of our two mutualists, and on the y-axis here with the growth rate of each of them. So again, we see this sort of Lee effect by stability. All right, so what happens if we're at this good cross-feeding productive equilibrium? We introduce a cheater who doesn't produce either, but is a little bit better competitor. Oh, sorry, this is just to show the dynamics of the symmetric mutualists lead to either one or the other equilibria. All right, so now we're introducing our cheater. So our cheater is now on the y-axis of this phase plane. And what you see is that the cheater basically connects this formerly stable equilibrium of the cross-feeding consortium doing great, and like connects it up, jumps over the unstable equilibrium, and draws us into the 0-0 equilibrium, which is essentially everyone's dead. So this is what we say when the cheater invades and then it crashes the consortium. On the right, we have the corresponding time series where we start near this equilibrium, introduce the cheater, grows initially, draws the cross feeders down below their, um, unstable equilibrium, and then since no one's actually producing amino acids anymore, they all die. So there's a problem, right? There's a common problem in mutualism theory, how to stabilize it against cheaters. And so one of the common notions on how to stabilize the system is to introduce space. So with that, in a project led by former postdoc Simon Stump, we put the same type of interaction into a stochastic spatial model on a lattice. And so the same kind of rules apply, but now we have localized interactions. So now you're only cross-feeding with individuals within some neighborhood and localized dispersal. So microbes only disperse to nearby sites. And so these are going to be the two big parameters here, the interaction distance and the dispersal distance. Okay. All right, so here we can simulate the system with intermediate range interactions and intermediate range dispersal. And the two cross feeders are going to be blue and green. The cheater is going to be in pink. And the empty site is white. So let's hit play and see what happens. So they're doing great. Then at some point we introduce the pink cheater. And you see the cheater invades. But in fact, it never excludes the cross feeders. There's a sort of, I'm just calling it spatiotemporal chaos. We didn't test that. But we have this dynamic mosaic where the cross feeders can do well in empty sites, but then they're continuously followed and chased out by the cheaters who crash them. But nevertheless, at large enough scale, they coexist. So this next scenario, we're considering. Oops, that's the same scenario. The next scenario, we're considering short-range interactions, but long-range dispersal. Okay. And if we run this one's a little bit more boring. But we see that in fact, the all three species manage to persist, so the cheater is not able to crash the system here. And I'll explain why this works in a second. But before we do that, we'll have yeah, always in the same one. Okay, so now we have long range interactions, but short range dispersal. And this is going to lead to some sort of pattern formation. I'll remember like when we learned about this, it was Friday night, getting ready to go home for the weekend, and Evan Johnson, Simon Stump were working on this project. They're like, "Hey, you got a minute? Got to show you something cool." I'm like, "Okay, I got a minute for something cool." And that's what they showed. So basically, under these circumstances, we see that the two cross feeders self-organize into these regular spatial patterns. In this case, they end up being, these parallel stripes. And this manages to exclude the cheater from the system. Now, a similar simulation, slightly different parameter values, shows that, again, we see the self-organization of the cross feeders. Now the cheater manages to persist, even gets in there and kind of messes with their pattern. But you can kind of make out here that the cheater, the only place it can make its living is at the interface between these more or less monospecific patches of the two cross feeders. And that's going to be key to understanding these spatial mechanisms to keep the cheaters out. And so this is an overview of the outcomes as a function of the interaction distance and the microbial dispersal distance. So up here in the corner, which is kind of like a well-mixed system, like our ordinary differential equations, we see that the whole system crashes. But anywhere along either of the axes here, we see that the system persists. Sometimes the cheater is actually excluded. Sometimes it coexists with the two of them. All right. So how do they manage? How does space save the day? I think in the case of the spatiotemporal chaos, it's pretty obvious, but the other cases maybe not so much so. So here's a case of the short range interaction, but long range dispersal. And so each microbe is only interacting with its nearest set of neighbors. And so what happens is if you're the cheater, you need neighbors of both types to provide both types of amino acids. But you might just get unlucky. You might be in a neighborhood where you're only getting amino acid 1, but not amino acid 2. So based on that, this is enough to keep the cheater out. We see the same thing in the case of the pattern formation, where the only place the cheater can obtain both resources is at the interfaces between these stripes. If it's like right in the middle of one of them, then it's getting the blue amino acid, but can't lay its hand on the green one, can't make it itself, so it's excluded. All right, so this is wrapping up part one of our older work from our DARPA project. So cross-feeding can promote species coexistence, can lead to multiple stable states. In well-mixed systems, it's susceptible to invasion by cheaters who can then crash the whole system. But in poorly mixed spatialized systems, cross-feeding can be stabilized by three sort of distinct ways: the spatiotemporal chaos, the uncertainty of not knowing who you're going to interact with, or through the self-organized pattern formation, both of which, basically prevent the cheater from being able to get both resources. All right, so we had a few other theoretical extensions to this work, which I'm not going to talk about today. So we introduced like a totally self-reliant species who produces both, try and investigate the Red Queen hypothesis. We looked at sort of an eco-evolutionary dynamics where we made resource synthesis not an either or, yes or no, but into more of a quantitative trait, which you can read about in these papers. But that was it. That was the end of our DARPA project. And so we quit working on cross-feeding for a little bit. Um, but then, it was in email correspondence with Ophelia Venturelli, and she mentioned that her master's student, Tyler Ross, had made such a cross-feeding system in the lab. And not only that, it exhibits some very interesting dynamics. And would I be interested in collaborating with them, help them with their model that they had? And so, of course, I said yes. And so, after a little bit of tweaks to the model and a whole lot of analysis, I think we have a pretty good understanding of what's going on in their experiments. And I should mention, um, Ophelia was a postdoc on one of these DARPA-funded projects. So I met her at our program meetings, back in 2017 or so. Since then, when we started working together, she was an assistant professor at University of Wisconsin, and she's now an associate professor at Duke. Okay, so here's the experimental system. They took some E. coli and they knocked out genes for synthesis of two essential amino acids. So this blue guy cannot make tyrosine for itself, and this orange guy cannot make phenylalanine for itself, but they do excrete. And luckily, like in our model, they're competing for glucose. And they have, fluorescent reporters so you can see who's who when you're to quantify them. This plot on the right is just experimental verification that they are in fact benefiting each other through cross-feeding. So it's comparing their abundance in monoculture versus in co-culture. And so in co-culture, their abundances are much higher. So we see that there's this cross-feeding going on. So Tyler's experiments were in a regime called serial batch culture. So basically, you would inoculate both species into a flask, grow them for 24 hours, um, passage a small fraction to a new test tube and give them a whole bunch of nutrients again, and repeat day after day. So this is serial batch culture. And so here's an example of the types of dynamics they saw on the right. And what's going to vary between these is the supply of external supply of amino acids they're given. And what we see here is kind of a discrete time system because they're only able to assay them once a day. So on the right, we see that, yes, the two guys coexist. Okay. Even if you don't give them amino acids in their media, they manage to persist through this mutualistic cross-feeding. That's good too. What happens in the middle? What happens in the middle is the big surprise, right? So instead of going to a stable equilibrium, we see this long-term period two dynamics here. So there's a period two cycle that emerges. So first thing you might be able to see here, this amazed me. There are replicates here, so there's actually two lines for each color, so the two replicates behave more or less the same. Um, so what's going on with that two cycle? Maybe it's something just about this set of input concentrations, but no, it's actually robust. So they also did some experiments where they more broadly varied the supplied concentration of amino acids. And you see that over a broad range of parameter values, we see something like this period two cycle. So where is it coming from? And how come our previous model didn't produce any cycles, right? So there's some missing ingredient here. And Tyler and Ophelia had the idea already, what was the missing ingredient, which is some sort of metabolic interplay between the two species. So in our original models that I presented before, the two species are on autopilot. They just, they excrete the amino acid that they make all the time, nonstop, no questions asked. But in reality, that's not actually true. So what we think is the case is that the rate that a species synthesizes the amino acid it can depends on its glucose supply. Okay, so here's delta tier A, and who can make phenylalanine but cannot make tyrosine. So in this case here, we see that it's going to be glucose limited on those hexagons there. And so they'll take up these to match their needs. And then the assumption is they're gonna synthesize the amino acid they can, phenylalanine, to match the glucose supply that they took up. And then we're gonna apply Liebig's law of the minimum to determine their growth rate. Here the growth rate's 2, but they only made enough phenylalanine for themselves, right? They don't have any extra, so they don't excrete anything. The converse case, if this guy instead is limited by the amino acid that it can't make, it takes up all the amino acid it can, it's greedy, takes up all the glucose it can, it's greedy, and then it synthesizes phenylalanine to match the glucose that it took up. So synthesizes 3. Unfortunately, its growth rate is limited by the amino acid tyrosine, and so it's got this extra glucose, it's got this extra phenylalanine. Extra glucose it can just burn off, but the extra phenylalanine, they don't want that, so they spit it out. And so this is the case where it's excreted. So to summarize this missing ingredient here, the rules that they follow is if you're limited by amino acid, you excrete the amino acid, the surplus amino acid that you synthesize. Whereas if you're limited by glucose, you just make enough that for what you need and you don't excrete anything. And this kind of makes sense because, I mean, these guys in their wild type, they can synthesize both tyrosine and phenylalanine. They usually don't have to worry about not having enough of it. So that's why we think that their synthesis is tied into their glucose income. All right, so this assumption was verified in the lab. So Tyler ran some assays, 24-hour assays, where he ran each species by itself. And gave them different doses of amino acids and glucose, and then measured the availability of those resources over time. And so what you see here in the upper left, we've got tyrosine is glucose limited because we gave them lots of tyrosine. They consume all the glucose. We got a little tyrosine left over, and they don't really excrete a lot of the phenylalanine. Whereas on the right, when they were given lesser amounts of the amino acids, then they basically don't run out of glucose. They do run out of amino acid. And then this is the time where they're spitting out and all that extra phenylalanine that they're synthesizing for nothing. And the same thing is true for both strains. So I'd say that this assumption is more or less verified in the lab. Okay, so now we can include that into our model. slight modification of our original model, where now instead of having that synthesis on autopilot, we compare their um, growth rate on glucose versus their realized growth rate. If they've got extra, they excrete it. If they don't, they don't. So this little plus means that basically it's the um, if it gets negative, then it's basically zero. So this is the model. And so can it recreate those period two cycles that they saw in their experiment? The answer is yes. Maybe you guys noticed this when I first presented this slide, but the solid lines here are experimental data, and the dashed lines are the model results from the best fit model. So again, you can see over here in the center, we have this period two cycle emerging from the model where it should. All right, so this is great, but it's a little bit of a question because the system is subject to this periodic daily dilutions. Maybe that's where the cycles come from. Maybe it's not internally driven, but it's something to do with the periodic dilution. So to answer that question, we took the same model and just ran it in continuous culture chemostat mode, where instead of having daily dilutions, they just had a continuous supply and dilution rate coming in. And what we found was indeed that there's a large region of parameter space that leads to these limit cycle type dynamics. Now, it is a little bit complicated though, because it's like 5 equations. We want to come to a more complete understanding what drives these cycles, under what conditions do we expect them. So to answer that, we tried to really reduce it to the bare minimum, to really strip it down. So we considered what we can relate a minimal model, and we made a bunch of assumptions for convenience, some based on numerical results. So first of all, we're going to focus on no external supply of amino acid. We just assume, based on numerical simulations of the full model, that the glucose concentration and total abundance of our species is roughly constant. We define then the relative abundance of the two strains as F, and yeah, we switch to linear functional responses. So now we've got this thing reduced to this three-dimensional system for the two amino acids, plus the relative abundance of the two species. And furthermore, we notice based on the parameter values that we have, is that there's a sort of separation of timescales where the resources are a fast system and the relative abundance is relatively slow. So we looked at the fast resource subsystem by itself as the function of the relative abundance, the slow dynamic variable. And so in this case one, depending on the relative abundance of the two species, either you have one resource totally depleted, the other one totally depleted, or both kind of there. But this always leads to a stable equilibrium, so no population cycles in this case. However, there's another case where these equilibria cross in a different way. And so now for intermediate relative abundance of the two species, the system is bistable. There's two different resource equilibria. Either resource two is at high abundance and resource one is depleted, or vice versa. And we can finally determine the conditions under which this holds. This is the full expression, but a necessary condition is this relationship between the stoichiometric coefficients. Okay, so this is the fast resource subsystem. Now let's introduce the slowly varying relative abundance F. So now instead of taking the relative abundance as a parameter, we'll make it a slow dynamic variable. And basically what we see are these relaxation oscillator type dynamics. So if we're at this equilibrium up here, species one is essentially being fed by species two. And so its abundance increases over time, but as its abundance increases over time, the availability of the resource it needs is going down until we reach this bifurcation point, in which case we jump off and get on the alternate equilibrium. And once we're on this alternate equilibrium, now the other species is being fed, and the relative abundance slowly tracks back down until we jump off the bottom again. So it's kind of like a van der Pol relaxation oscillator. This is what the dynamics look like in that case, but we can also look at this in cartoon form. So, and the way I kind of think of it, it's not really reciprocal altruism, but it sort of feels like it. So at the beginning, so the size of these microbes over here is going to denote the relative abundance. So we're going to start with them equally balanced, but we're in the state where the orange guy is feeding the blue guy because he's limited by amino acid. This guy ends up limited by glucose, so he doesn't excrete anything. So basically, the orange guy is doing all the work here. And so the abundance of the orange species goes down, the abundance of the blue species is going up. But at some point, the abundance of the blue species is too big, the orange one's too small, can't feed all those blue guys. So then we see that switch. So that's going through that catastrophic bifurcation. And now they switch roles. So now the blue guy is limited by amino acid. So he produces amino acid for the orange guy. The orange guy is now limited by glucose, so he doesn't excrete. So he quit working. And so we get this cycle going back and forth over time until it repeats and keeps going around the circle. All right, so that's kind of our understanding of what drives these oscillations. I've got two more little bits here. One is getting back to this theme of how can we stabilize this interaction? How can we make it robust against cheaters? And we saw earlier that spatial dynamics could provide an answer. How about temporal dynamics? So this just shows in the case of a stable equilibrium, the cross feeders are doing fine. At time zero, we introduce this green cheater. As before, the green cheater increases in abundance, crashes the system, everyone goes extinct. But now if we change the parameter values, change these stoichiometric coefficients to get into our limit cycle regime, now we see that the oscillations amongst the cross feeders actually keep the cheater out. So we can understand that by looking at the growth rate of the three species. So the two cross feeders alternate between being fed and feeding, but the poor cheater, right? He's only got like a few minutes here where he's got enough resources provided by both. Most of the time, he's limited by one resource or the other. So this is enough to keep the cheater out. So it's kind of a temporal analog, what we saw in our spatial models. And so finally, there's a question that came up from one of our anonymous reviewers. And so reviewer two, if you're in the audience, thanks for this question. They were interested in how do we connect the continuous time limit cycle that we analyzed with the discrete time period two cycle that Tyler and Ophelia saw in their experiments. So how do we connect these two? And so the first thing we need to do is crack open the black box here. So experimentally, they only measure these species at every dilution, but with a model, of course, we can keep track of them in between. So the first thing we do is look at the continuous dynamics between dilution events. And this is really what the two cycle looks like. And then basically we just modified our models to adjust the period of the dilution, but at the same time change the size of it to maintain the equal total input of nutrients and dilution. Okay. So we're going to start with very frequent small dilutions. And so on the left, we have our idealized limit cycle. On the right, we have what happens with 1 hour, and there's a 15.23 hour limit cycle. So if we dilute them very small amount every 1 hour, we get basically what looks like a fuzzed out approximation of the continuous limit cycle. Technically, this appears to be aperiodic. But in the grand scheme of things, it's pretty much like the 15.23 cycle. Same thing now for less frequent, bigger dilutions. Again, we're just fuzzing out that limit cycle more, and we get trapped onto periodic attractors, and they go in and out of fashion. So here we're on a period 8 cycle, where the forcing is 2 hours. 2 times 8 is 16, which is pretty close to 15, so kind of locked in there. And as we increase the period of dilution, this fuzzes out more and more, going through a lot of complicated dynamics. But it's still the same general character. But at some point, when the dilutions are getting big enough, then we're kind of moving into a new regime. These guys are getting down to low abundances after every dilution. Takes them some time to rebound. Um, and finally, we connect up with our 24-hour cycle that was matching the experiment, which I think is really interesting here is if instead, the experiment was done with 30-hour forcing instead of 24, we would have never noticed anything 'cause it's just a period 1 cycle. We wouldn't even investigate this. This is the summary of all that, summed up in this bifurcation diagram as a function of the forcing period. So here's the experimental 2 cycle. These are the minima, no, the maxima of the force model. It goes through a lot of bifurcations, gets really crazy down there as we're approaching the limit cycle. But the big picture is that these things more or less connect. All right, so to conclude part 2, this metabolic interplay mediated by this glucose-dependent excretion can lead to novel oscillatory dynamics both in the lab and in the model. These population cycles don't rely on external forcing through the daily dilution, but emerge through the internal dynamics. So we think this may be one of the first examples of a mutualism-driven population cycles. I mean, we all know about predator-prey cycles or intransitive competition. So mutualism has now joined the party as a possible mechanism of population cycles. We dissected this as a type of relaxation oscillator. These population cycles can prevent invasion by a cheater, just like the spatial ones can. And finally, there's a potentially complex interplay between this external periodic forcing and the internally generated cycles. So with that, I'd like to thank all of the co-authors on these papers, as well as funding sources that helped out with that. And thanks for listening, and be happy to take any questions.
Thank you very much, Chris, for the excellent talk. And if you have any questions, please raise your hand in the participant list, and I will be happy to call on you. So any questions? Yes, Sharp, please go ahead.
Hi, Chris. Can you hear me?
Yep.
Okay. Thanks a lot for the presentation. It was really nice. One quick naive question, just right at the end. I didn't really get why for a 30-hour period you wouldn't notice the oscillations, because I thought that after 24 hours they converge to equilibrium, so it will be the same thing for any longer period.
Yeah,
Don't
it
know.
just turned out that for longer, for that, say, 30-hour forcing, they get trapped on like a period 1 cycle. And so then experimentally, like when they're in the lab, just every day they measure the concentration after dilution. It would be the same thing every day. So you wouldn't even suspect that there's, potentially interesting dynamics going on underneath.
Okay, but is this related to like some of the the dynamics of the resources that we don't see in that plot?
Yeah, I mean, definitely the resources are involved. I mean, I guess it's just a different regime. So I guess
Okay.
yeah, I mean, at some point, right, if you dilute so infrequently, they just deplete all the resources, and then they're just kind of
Okay,
hanging
okay,
out,
that's a good point. Yes.
waiting for the
Yeah,
next day. So
sure, I see it. Thanks.
yeah,
Thank you. Prajval, please go ahead.
Hi, thanks, Chris.
thanks.
In the model, if there is, or not necessarily in the model, if the amino acids are not limiting, I would expect they're only competing for glucose. So the model should say that one of them should go, or one of them should be excluded. But I don't know if the slide of the experimental parameter scan was showing any sort of exclusion. I mean, this could be because they're feeding off something else, but I'm wondering how much of this would change the results of the oscillation.
Yeah, let me see if I can find that share screen one more time. Sorry, resizing a window here. So yeah. Yeah. So, if they don't, if the amino acids aren't supplied, then exclusion's impossible. But if the amino acids are supplied, then in fact, if you supply a lot of tyrosine, at some point, like, yes, it becomes competitive exclusion. So there's little bands of competitive exclusion along this figure, but it requires external supply.
Mm. Yes, but I mean when both are in excess supply, so they're not competing for either of. I mean, there's no point in releasing or leaking the amino acids anymore. So they should
Mhm.
only be competing for glucose then.
Yeah, I mean that would probably be a supply point up here,
Yes.
right? Where yeah, the amino acids we're dumping in tons of them, so they don't need to worry about amino acids. And you
Mm.
see that's in this little band here labeled delta phi A extinction.
Okay. Yeah.
Yeah.
Perfect. Thanks.
Thank you. Angie, please go ahead.
Hey, Chris, super cool stuff. I still don't understand something about the cycling bit. You have an ODE, right? And you're getting like different period cycles out of it. Are you pulsing it then? How do you,
So
yeah.
with the ODE, without the forcing, there's just a single unique limit cycle.
So okay, so you have to force it to get this connection where you have your different period cycles.
Yeah, exactly. So in this, see if I can find this thing. Yeah. So over here on the left, this is like the chemostat mode, and it's just this like nice idealized, beautiful, 15.23 hour limit
Yeah.
cycle.
So what are you
Um,
forcing in the model? I guess I
what's
don't understand.
that?
What are you forcing in the model?
So the forcing matches the serial batch experiment. So then basically every period, every tau, we extract out some fraction of the media
Oh, okay.
along with everyone who's in there, squirt that out and then refeed them with like fresh medium, but like with no one there.
But
So
no,
it's both
okay.
like it's both a supply of resources, but it's also like a mortality event.
Okay. Okay. And so you have this discrete pulse
Yeah,
ODE.
yeah.
That's what you
In
spit
this
out.
last part, yeah.
Okay. Okay. I missed that.
Hmm.
And if I can ask one other quick question for the stochastic stuff, when was there any way for the recruitment to happen stochastically, or is like if they died out, they were gone from the system?
Yeah, that's a good question. No, it's just a closed system and no one's immigrating or anyone. So you're right. Probably, if I ran this for like to the end of the universe, they would all die. Um, but they managed to persist for a long time.
Okay.
Yeah.
Thank you.
Thank you very much. Ayush, please go ahead.
Thanks for the talk. I think this is a relevant slide for the question. So do you have something idea about the relevant experimental system to test the ideas which you have from the spatial structure on the dynamics? Because I guess in the experimental part, the setup was mostly a well-mixed population, right?
Yeah. Yeah, I mean, I'm not an experimentalist, so I
Right.
can't say for sure, but I imagine you could grow them in Petri dishes on some kind of agar, and that would yet. I mean, you'd have to think about how you would manipulate the dispersal and the diffusion of resources. So that might not be so straightforward.
Right.
Um,
Right. Okay.
But I think people do experiments with bacteria on plates, right? So I think something like that.
Yeah, but I always feel some sort of disconnect, especially when it comes to space between theory and experiment. But
Yeah, I mean, obviously, they don't live on like this grid, right? They're in there and they're kind of like pushing each other out, and they're like maybe getting into the third dimension. So
right,
it is definitely a idealization.
right, right. Thanks.
Well, thank you, Gisa. Please go ahead.
Chris, thank you very much. It's very interesting, but can you go back to the slide in which you explained what are the spatial effects which defend against the cheaters?
Like here, maybe, or here.
No, no, you have had a list of, I think, three
Oh
effects.
yeah. Uh-huh. Yeah. These.
Yeah. Okay. Yes. If So intermediate range of interactions and dispersal. So both of them. Uh, 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 what is the difference between them? It's not.
Yeah, so I mean, you can kind of see it in this figure. So these
Yeah.
ones, I mean, of course, they're not animated, but you can kind of see like, these ones here, we have these kind of big blobs
Yes.
of different colors. And if it were animated, they'd be chasing each other around. So this would be kind of the spatiotemporal dynamics where you have these large patches, but they're not static in space and they're running around. Down here with the pattern formation, once these stripes set up, they're more or less going to stay like that for a long time. So these don't move around. And then the cheaters, if they're going to be in the system, they're kind of restricted to the interfaces. And up here with the neighborhood uncertainty, it looks kind of boring, but basically the two cross feeders are more or less tightly interspersed with each other. And this will it'll change a little bit at random over time, but won't have these big coherent blobs of species chasing each other. So,
Yeah.
I mean, it's kind of, I mean, yeah, it's there's no hard and fast, like breakpoint between the two, through the three regimes. It's just kind of like we wanted to label them somehow.
Yeah, because there are two variables and two distances, and either of them should be limited. And
Yeah,
the best, okay, this is
yeah.
the
And then once we get up here, and then we're in the well-mixed system more or less, and then the cheaters manage to like crash the whole thing regardless.
yes, yeah. I'm asking this because I have colleagues who made similar simulation, not in these systems, but especially against cheating.
Yeah. I mean,
Yeah.
certainly it's not an original idea.
Yeah,
For
well,
sure.
of course, but this essentially the very same. So my understanding that if the research diffusion is limited, if the microbe dispersion is limited, it means that the cooperators, not the cheaters, tend to remain near to each other, and research distribution distance means that if it is if the resource has a limited diffusion, then the cooperators overrepresented in that small region where it is present. This is
Hmm.
this is my understanding of
Yeah.
these two. Okay, thank you.
Thanks. Neil, please go ahead.
Hey, yeah, I was curious about, and you might have mentioned it, sorry if I missed it, but how much parameter fitting is going on in here? Are they pretty stable for these different simulations that you got going on, or are you doing some
Yeah.
fitting for every
Yeah.
one? What's going on?
Yeah, so in the first part, it was blissfully free of real world parameters, so we can ignore that part. In the second part here, with Tyler and Ophelia's experiment, yeah, I think they fit, to the whole, all the experiments together, got one set of biological parameters, and then just fixed it for the rest of the analysis. Um, so that's why, this one perhaps looks really nice match. This one not perfect. I mean, I guess so if we like refit it just on this time series, it could potentially be better, right? But I think they fit it on the whole, everything at once, and then applied those same parameters across the board. So it seemed like a relatively fair way to do it.
Yeah, so when you say fitting it on the whole thing, you're talking about like how many parameters per species, or what are we talking about? What's being
Oh,
fitted?
that's a good question. Somewhere I've got like the table. Let me see if I can pull that up.
Oh, you could just tell me if it's a bunch of stuff or just a few things.
Um, It's definitely some things. Basically I mean, here it is. I can switch over here. So from the depths of the supplemental material. Yeah, I mean, each species has a maximum growth rate. We've got half saturation constants, these stoichiometric coefficients. Um, no, this is for a slightly different, sorry, that's the wrong model. That's for the original one. Here's the one that we used. Yeah, so we got maximum growth rates, a handful of half saturation constants, quotas, and then everything else is under experimental control, like the dilution rate and the nutrient input. So I
And
guess
that's for
that's 12,
is that
12
tw
parameters. Yeah.
for two species or three species going on there?
For two, yeah.
Okay.
There's just I think the experiment's mostly focused on the two cross feeders and not the cheater.
And those are pretty stable for the different results that you got. Got it.
Yeah, I mean,
Nice.
we just use this same set of parameters for all those results essentially.
Yep. Got
Uh,
it.
we have an idealized set for making nice figures too, when we're in more like heuristic mode.
Right. Okay. Congratulations. Thanks.
Thanks.
Thank you. Rafa, please go ahead.
So my camera's not turning on, but you guys can still hear me, right?
Yep.
Okay. So Chris, your answer to one previous question about the reason for the oscillations in the differential equation model. So I think I might have then misunderstood at first. So I thought that you saw oscillations even in the chemostat model. Well, let me see if I understood the answer to the question. You said that the reason you see the oscillations in the ODE model is that you're forcing the dilutions. But I
Yeah,
thought that,
no.
okay.
No. So yeah, we definitely saw, nice, oscillations even in the chemostat
Mhm.
unforced model. And that's that, relaxation oscillator kind of mechanism that I was talking about. But it was like, yeah, I mean, that was pretty much how it was in the paper until, reviewer two asked about connecting the
I
two.
see. Right.
Um,
So my understanding of why the oscillations were happening is because of that discrete change in behavior of
Yeah, there is a discrete change in behavior, but it's not due to discrete forcing. It's this sort of like relaxation thing. If I can find this. Yeah, where, basically the relative abundance here is on the x-axis, which changes slowly, and then the availability of the two resources is on the y-axis. So, as one of them is feeding the other, um, the one who's being fed abundance goes up. The one who's doing all the work, their abundance goes down. And at some point, there's a little bit of a revolution, and they switch roles and go back the opposite direction.
right. So we need this kind of discrete change in behavior. Like I start making the amino acid because now I'm limited by a different resource. So if we try to smooth that out, right, and didn't have
Hmm.
that type of jump in behavior,
Yeah.
could we still get
Um,
the oscillation?
I think you could, I mean, so the sharp pointiness in a lot of these figures comes from Liebig's law of the minimum, right? So it's this piecewise continuous differential equation, but it is continuous, right? So it's not like discontinuous equation. So I think that if we were to smooth this out, and replace Liebig's law of the minimum with something a little bit more interactive, then, it would just kind of like round this corner out a little bit. I mean, you definitely need this sort of saddle node bifurcation at each end. But I don't think it depends strictly on the, sharpness from Liebig's law of the minimum. In fact, Tyler's original model, well, maybe it had Liebig's law of the minimum in there too. But it was a little bit smoother and it still did the same thing.
Okay. I had one more question about the spatial stuff. So how exactly did you model the dispersal part of the microbes? Were they acting like, coral or like plants that just send a seed somewhere
Yeah, I
and
mean, it was like
sprout somewhere?
we're back in the 90s doing these spatial stochastic models where,
Okay.
they're dispersing, they have to land on empty site, otherwise it's a waste. So
Okay. So like a forest and they only see resources in their vicinity and the resources diffuse.
we call it resource diffusion, but it's actually more like they interact with everyone in this interaction neighborhood. So that's who's potentially like, providing them with nutrients. We don't model the resources explicitly here.
Oh, interesting. Okay. So the resources are factored out. Like the way you
Yeah.
do that is what separation time scales. Like how do you?
Um, yeah, I think I'd have to review this
Okay.
Stump et al. Paper, but the details should be in there.
Okay.
Yeah, so it's not a complete like one-to-one match between our original ODE model and this stochastic spatial model. Because we don't have glucose in here, we have empty space, but I think it's the same essentials.
I gotcha. Okay, great. Thank you.
Thank you very much. Any other questions?
Can I ask a final one?
Of course.
Yes, still about the spatial model. So have you considered studying this system on evolutionary timescales? Because you could think that the resident species could evolve, for instance, the dispersal, and you could imagine that after many iterations, they could select for lower dispersal because that's more robust to invasion by cheetahs.
Yeah, that's an interesting question. We did have one model over here that was more of a sort of, eco-evolutionary dynamics, where we made the synthesis a quantitative trait subject to, selection. And that was pretty heavy, ended up in Journal of Theoretical Biology. But yeah, we didn't think about the evolution and dispersal. That's a good question. That would also be interesting.
And just to make sure, cheaters and residents have the same dispersal, right? In your model?
Yeah.
Okay.
Everyone's got the same.
Okay. Thank you.
Thanks. Any other questions? One semi-random question from me. When you were talking about the fact that the oscillating dynamics excludes the cheater, you showed a graph of the growth rates.
Yeah.
Illustrating the mechanism.
Yeah, let me see if I can find that again. Oops. Yeah, here.
That's the one. What I'm a little confused by, but maybe it's just a plot property, is that those growth rates never drop negative.
Yeah, so that's a good point. So the growth rate, the colored line is sort of their birth rate, and then this dashed line is their death rate due to dilution. So basically, in this period, the orange guy is feeling great, the blue guy is suffering, and the cheater suffering. But when they switch, now the blue guy is feeling great, but the cheater is still suffering. So the only time the cheater has like, an opportunity is that brief switch over moment when there's both resources.
Thank you. Okay, any other questions? If not, then thank you again, everyone, for coming, and stay in touch. I think in 2 weeks we'll be back, so we'll all hope to see you there. So thanks again, Chris. Bye,
All
everyone.
right, thanks.
Have a
Yep, bye.
rest of your day. Bye-bye.